Okay. Yeah, so you're talking about sodium nitrate. Okay, 16% of nitrogen, the rest all is sodium. Okay, and it is why it is usually used is because apart from soil, it can be also applied to plant, small ornamental plant dressings and all. So it helps in the growth of that particular region. Okay, and basically helps in the aesthetic value of the plant. Okay, that is your sodium nitrate. Apart from that, okay, now sodium nitrate is used as in these plant dressings and all, but not preferred when it has to be used in the soil, because the soil cannot hold sodium nitrate. It's quickly leached out. Okay, fine. Then there is ammonium sulfate. Okay, ammonium sulfate is another one which is a white crystalline salt. Okay, now it contains about 20 to 20% of your nitrogen in the form of ammonia. Okay, or ammonium ions basically. Now apart from it, there is also ammonium nitrate that can be used. Okay, then there is ammonium sulfate nitrate. Okay, then there is ammonium chloride. Okay, then urea is the most commonly used one. Okay, then there is calcium ammonium nitrate. Okay, calcium ammonium nitrate will act as a compound fertilizer also, because it will also provide calcium. Okay, fine. Then we come to your phosphatic fertilizers. Okay, now phosphatic fertilizers are basically those, okay, which contain phosphorus as their primary component. Okay, there are two. There is superphosphates and basic slag. Okay, two types. Superphosphate and slag. Okay, superphosphates are those which contain high amount of phosphate. Okay, now example of superphosphate is calcium dihydrogen phosphate. Okay, now it is the most, and why the superphosphates are usually preferred is because they are not plant specific. I mean, see, sometimes what happens to fertilizers also, when you have to apply these chemical fertilizers, they may not be suitable. It's like an allergic reaction. Okay, some plants may be not suitable to certain. Okay, but what is the advantage of superphosphates and why they are widely used is because they can be used for all varieties of plants. Okay, they have not been known to affect any plant adversely. Okay, then basic slag. Slag is used when in regions where your farms are closer to factories. Okay, like steel factories. Now slag is a byproduct of your steel factories. Okay, as in your steel factory, when the iron ore is formed, okay, from the iron ore you are going to extract iron. Now this iron ore, okay, contains phosphoric acid. You use this, okay, for your plants. Okay, it provides phosphate back to the plant. Okay, and this, the byproduct is known as slag. So it is a readily available form because it's a byproduct. The industries will give it for free. Okay, and easy for farmers whose farms are near these. Okay, fine. So this is also used as a second type of phosphatic fertilizers. Then is potassic fertilizers. Potassium or potassic fertilizers are not usually used in India. Okay, because in India there has been hardly any case where soils have been deficient in potash. Okay, or potassium. Mostly potassic fertilizers are used in outside countries like US and all. Okay, India mein usually potassic fertilizers are not used. So now potassic fertilizers, okay, there is two, muriate of potash and sulfate of potash. These are the two forms of okay, sulfate of potash, the name suggests, like potassium sulfate. Okay, fine. Now muriate of potash, okay, it is a grey crystalline salt. Okay, it contains around 50 to 63% of potash. Okay, and it can be, okay, it is highly soluble in water. Okay, so it is present in this form, K2O. Okay, highly soluble in water, so it usually is given in the form of liquid, okay, and that's why it's known as muriate of potash. Okay, muriate is usually a term used when anything is there in water form. Okay, anything which is going to be saturated with water, something is known as muriate. Okay, then coming to your fourth, okay, that is your compound fertilizers. These are your basic fertilizers, okay, but when we use a combination of these, okay, they become your compound fertilizers, okay. Like compound fertilizers are those wherein multiple nutrient materials are supplied, okay, supplying 2 to 3 compounds, okay, together. Now, for instance, if a soil is deficient in both nitrogen, okay, and phosphorus, then we should use a fertilizer which provides both these components, right? For example, amorphous, okay, this is a very widely used fertilizer, okay. Amorphous contains around 16% of nitrogen, 20% of phosphoric acid. Nitrogen, 20% of. So both provided, okay. Then apart from this, there is also monoammonium phosphate, okay, which can be used. Then there is also diammonium phosphate which is used. Okay, this was your compound fertilizers, wherein you use a compound which is providing both the nutrients. Okay, mixed fertilizers is when you are combining or adding a mixture of fertilizers. Okay, getting the difference between compound and this. Compound is when one single compound is providing both its nutrients. Mixed is when you are using a mixture of it. Okay, so mixtures which compound fertilizer, yeah. So now sometimes these mixtures, okay, will contain, okay, all the three, like nitrogen, phosphate and potassium. Okay, you will use compounds which contain all three, and that mixture is known as a complete fertilizer. Okay, because it is providing all the essential nutrients. Okay, that was about chemical fertilizers. Okay, so we just had to know the basic 3 compounds. Okay, that is nitrogen, phosphate and potash. Okay, and then mixture of these or one compound providing both these. Okay, if we have chemical fertilizers, then why are we shifting to biofertilizers? Okay, one very big problem, okay, of chemical fertilizers has always been that even though you, supposing I apply 10 kgs, okay, of fertilizer to a farm, the yield that I am getting is hardly worth 2 kgs. So first of all, they become expensive. Okay, chemical fertilizers are expensive, so sometimes inaffordable. And even though a farmer does spend so much, okay, to get in, the yield that he is getting is very less. Okay, so coming to biofertilizers, our main aim is to solve 2 things. Okay, first to make it more affordable, and second to increase the yield. Now, why this yield is reduced is because very few of the chemical fertilizers can be held in soil. Okay, like you add the fertilizers, okay, then continuous irrigation has to be done. During this irrigation, most of your fertilizers are washed away. Okay, these compounds cannot be held in by the soil, okay, or by colloidal particles in the soil. Okay, so then came the concept of biofertilizers. Now basically, biofertilizers, as the name suggests, has got something to do with biological organisms. So basically, when organisms are used as fertilizers, they are known as biofertilizers. Okay. Okay. Now we are going to study the basic or the most commonly used biofertilizers. Okay. First is your legume rhizobium synthesis. Okay, we've already studied just like some while ago. Then there is Azolla anabina symbiosis. Okay. Then we are going to study about loose association of nitrogen fixing bacteria. Okay. Then we are going to study about something known as diazotrophs. Then cyanobacteria, mycorrhizae, which are fungi, okay, phosphate solubilizing bacteria and fungi, and there is another interesting organism known as Frankia. Okay, so we are going to study about 8 types. Okay. Now coming to the first one. Okay. Okay, very easy this is. Okay, leguminous plants. Okay, they're cropped. It's like an alternating season. This is known as, this type of cropping is also known as alternative cropping. What you do is, like, once your harvesting is done, okay, of one crop, then you clear the farm, you grow these leguminous plants, you allow them to grow, then you get byproduct, like supposing groundnut. Okay, then you're getting groundnut production also. Okay, because if you grow groundnut plants, you will get groundnut production. Okay, then once their season is over, these are usually fast growing. Leguminous plants are usually fast growing plants. They will, nitrogen is come back to your soil, you remove these, okay, and then you plant your crop. Okay, so legume rhizobium symbiosis is very simple. Okay, your root nodules, they will again undergo nitrogen fixation and everything. Okay, alternative cropping is done. Okay, then is your second, that is Azolla Anabina symbiosis. Okay, forget Azolla, but Anabina, you all know what is Anabina. It is also nitrogen fixing. Okay, now Azolla Anabina symbiosis. Now Azolla is basically your floating freshwater fern. Okay, floating freshwater fern. Okay, Anabina, okay, it remains associated with Azolla in the leaf cavities of Azolla. Okay, again symbiotic relationship. Okay, Anabina fixes nitrogen, in turn gets its shelter in the Azolla leaf cavities. Okay, now this this Azolla plant, okay, it is a fern, okay, but apart from it, it also harbors an algae within it, small algae, azole that is called. Okay, fine. Now this algae fixes atmospheric nitrogen, okay, and releases nitrogenous compounds in its leaf cavities. Okay, how is it helpful to us? Okay, what we do is that we take this Anabina Azolla complex, okay, we flood our fields with water, okay, because it requires water for its survival. We flood the fields with water. This can be used in cases of rice, okay, where anyway your field is flooded, okay, and Anabina is exclusively mostly used only in case of rice fields. Okay, we first cause cause production of these in large tanks. Okay, then once your rice plants or seedlings start attaining a particular height, okay, you apply these. So nitrogen is being constantly there. Why nitrogen is required highly in rice fields is because, see, because of lots of water, okay, water becomes stagnating. So in rice fields, if you see, water has to be let out, a new amount of water has to be, this process of irrigation has to go on, okay, because of this leaching occurs to a high extent, okay. That's why you need biofertilizers or microorganisms which will not be leached away. If you apply any amount of chemical fertilizers, they are going to be leached away in the soil because of this huge amount of water. Okay, that's why Anabina is mostly used in rice fields. Okay, in the presence of water, they perform the normal, normal activities, and nitrogen becomes available to the plants. Okay. Then loose association of nitrogen fixing bacteria. This is as simple as it. We first grow bacteria in the laboratory or greenhouse, not greenhouse exactly, but basically you have a large repository, okay, or you cause reproduction of these bacteria, and then what you do is that you mix these bacteria in your manure, okay, and you apply the liquid manure all over your field. Bacteria go into the soil and they perform their normal atmospheric nitrogen fixation. Okay, easy. Okay, now that was your third one, free living bacteria. Okay. Now coming to the fourth one, that is diazotrophs. Okay, sometimes also written as diazotrophs. This is the same thing. Okay, it just some texts vary in their spelling. Okay, now diazotrophs are basically a large group of aerobic chemolithotrophs. Okay, 1 minute. Let's get the term. Chemolithotrophs. Chemical land trophs. What they do is basically from the ground, okay, or from the land, they will take in chemicals, and that's how they survive on those chemicals. The chemicals are their food. Okay, these are, yeah, chemicals is their food. Okay, lithotrophs is when anything related to food, autotrophs, heterotrophs. So trophs is when it is basically in terms of your food food consuming. Okay, now a very widely used example is your Thiobacillus desulfovibrio. Okay, this is your microorganism. Okay, Thiobacillus desulfovibrio. Okay, now these are free living. Okay, and what they do is that apart from just taking in or eating chemicals, they are also capable of nitrogen fixation in some cases. Okay, fine, because they also require nitrogen. If they are going to survive on chemicals, nitrogen is also chemical, some part of it. Okay, so they also perform atmospheric nitrogen. Okay, apart from it, what happens with these chemolithotrophs is that in case of what, like if there are rocks, okay, what these chemolithotrophs do is that slowly, like this is a very slow process, years and years, they will start breaking up these rocks to take their minerals. So what we do is that we place these small rocks within the soil. These bacteria continuously are breaking into the rocks, okay, taking in minerals that they require. The rest of the mineral that is left is available to the plant. Right? Fine. So these are also acting as biofertilizers. Okay. These are diazotrophs. Then there is cyanobacteria. I don't think I need to explain you what cyanobacteria do. Same old thing, nitrogen fixation. Okay. Now coming to mycorrhiza. Now this is, I think we've not studied this before. We're done with cyanobacteria. Sixth one is mycorrhiza. Okay. Now mycorrhiza can be ectomycorrhiza or endomycorrhiza. That is, they can either be on the surface, okay, or inside. Now what these mycorrhiza do, this is also symbiotic association with the plant. What these mycorrhiza do is they will obtain their food from the plant. Okay. In a way, they are acting like parasites, if you go to see. They are obtaining their food from the plant. But in return, what they are doing, okay, is that these fungi, okay, fungi, hyphae, can go along, okay, they can branch out. They will obtain these minerals from the soil, okay, and make it available to the plant. You're getting? They are not helping in nitrogen fixation or not having performing any function on their own, but they are helping accumulation of your minerals from the soil. Okay, fine. Getting it? And since they can spread large and they can go into depths also, so more amount of minerals available to your plant. Okay. Now these are usually found within your cells of your root, obviously. Okay, they go and lodge themselves within the cells of your roots. Okay, but unlike root nodules, they don't form nodule-like structures. That's all. That's the difference. Okay. Then there is phosphate solubilizing bacteria and fungi. Okay, same thing. What your diazotrophs do? Okay, that is they break down your rocks. Okay, but they take in or they absorb exclusively phosphate. Okay, if you put them in conjunction with plants, okay, they will make this same amount of phosphate. If they break down large amount of phosphate, they will use whatever they require. The rest will be available to plants. Okay, so this is used when you're in farms wherein your phosphate is very much deficient. Okay, it's like an alternative to superphosphate when you can't afford superphosphates.
Yes.
Yeah. Okay. Then I think phosphate solubilizing bacteria, if you want to know the examples, there is Aerobacter acrogens, then there is Thiobacillus, Bacillus, all these species do this phosphate solubilizing. Now coming to Frankia, this is what I wanted to explain you because I think this is new to you all. Okay, 7th one was phosphate solubilizing. Frankia are basically, type of actinomycetes. Okay, actinomycetes are also known as ray fungi. Okay, ray because they form a ray-like structure. Okay, the hyphae extend to form a ray-like structure. Okay, now some higher plants they form nodules with these Frankia. Okay, fine, getting it? Frankia they form nodules, as in not within the plant but externally. Okay, they appear like nodules. Okay, now what happens, these nodules, okay, these are also seen on stems of some aquatic plants. Okay, some aquatic plants also. Now apart, they do two things. Okay, they have dual mechanism. Okay, apart from also what fungi do, okay, like what your mycorrhizae were doing, taking in minerals and providing it, they can also perform atmospheric nitrogen fixation. Both. Okay. The only problem why Frankia is not used is that it is very specific in its association with plants, as in it can form association with only certain types of plants. Okay. That is why Frankia has not yet been used very widely. Okay. Now, yeah, that's all about your Frankia. Now, this Frankia is used mostly in terms when you want aquatic cultures. Okay, in those this Frankia is used as a biofertilizer because otherwise there is no other form of biofertilizer that you can apply. And also in some developing countries this has been used, but this is not a very widely used technology. Required, right? Macronutrients, micronutrients, and the forms in which they are available, and what happens when there's excess or there is deficiency of it. Okay, now minerals available to the plant in soil, okay, or by direct application or any of it. Okay, there is a certain process or method by which the plant takes it up, right? So today we are going to study the different processes, okay, involved in mineral absorption. Okay, we finished mineral plant nutrition. Now we are going to do the second part of plant nutrition, that's mineral absorption. Okay. Okay. Now there are two methods, okay, basic methods. We've learned about this before, okay, passive absorption, active absorption. Okay, under passive absorption we are going to do the different types of theories that came up, okay, and eventually the one which actually has been accepted, that's mass flow hypothesis, ion exchange diffusion theory, and Donan equilibrium theory. In active absorption we are going to do carrier concept and electrochemical gradient. Okay, now first coming to your passive absorption. Okay, what is passive absorption? What does the name passive suggest? There is no energy involved. Okay, when minerals are taken in just by simple diffusion, okay, as in from higher concentration to lower concentration or wherever required, okay, that is known as your passive absorption. Okay, now your first one is mass flow hypothesis. Okay, what is mass flow hypothesis? Basically what this theory states is that ions, okay, you know minerals, okay, are in the form of ions. They're taken in by a plant in the form of ions. Okay, so ions move along with the weight of water, okay, or with the mass of water. So what this theory is trying to say is that the principal method by which minerals are absorbed, okay, and distributed in the plant is only xylem, then because it has to constantly move with water. But we know that is not the case, right? Minerals are taken in by the roots also, and even sometimes by leaves and stem and everything, okay. So this was, what do you say, not accepted, okay. Then comes your ion exchange, okay. Now ion exchange is basically, what this theory states is that, like, consider this is your plant, okay, these are your roots, okay. Now what this theory states is that ions, okay, which are there in the soil, they become adsorbed on the surface of your root, okay. Now here's a root cell, supposing, okay. Eventually what will happen, this ion will enter the root cell in exchange with something else that goes out, mostly H plus or, okay, they are exchanged with any of these two, okay. So this, to an extent, this theory is true. We have seen that exchange of ions always takes place across the membrane, okay. Now there are two types of this exchange also, okay. One is your contact exchange, okay. Second is okay. These are the two types of ion absorption that takes place. Remember, we are still studying about passive absorption right now, okay. So basically no energy involved, okay. So now what this what contact exchange says, okay, is that ion exchange takes place, okay, because either by your ions, okay, coming in and getting dissolved with the carbon dioxide, okay, or there is a simple physical exchange, okay, like how I was explaining you, H going out and another ion coming in, okay. It could be your nitrate ion or sulfate, okay, anyone, okay. So physical exchange, that's what we are trying to say when we are saying contact exchange, okay. Now in terms of carbonic exchange, okay, carbonic acid exchange, when you are talking about carbonic acid exchange, okay, we know that carbon dioxide, okay, is released by the plant during respiration. During photosynthesis it takes in, but during respiration it releases, okay. So what this theory says, okay, is that cells, when they respire, okay, they produce carbon dioxide. This carbon dioxide mixes with water, okay, to form carbonic acid, okay. Then this carbonic acid which is formed immediately further breaks down to form this, okay. And then this is the one which gets exchanged. Same thing, but they're just saying formation of carbonic acid, okay. Getting the difference between the concept, okay. So basically when we're talking about contact exchange, we're not talking of carbonic acid, that means H plus ions could be produced by many of other metabolic processes, okay. So in a given plant you can say both of these also can occur together, yeah, both are partly responsible for ion exchange, okay. Now then comes your diffusion theory. Simple diffusion, okay. As and when your water is getting transpired, okay, there is a pull, okay. Because of this pull, your ions will start entering with the exchange of your other ions, okay. Now then is your Donnan equilibrium theory, okay. Now what Donnan equilibrium theory says, okay, what this guy Donnan said is that given any cell, okay, not just, we're not talking about plant cell right now, given any cell, okay, in the body or anything, this cell, okay, the cell wall or the cell membrane, okay, has an a balance, an ion balance occurring across it, okay, understanding ion balance, okay. So there is an electric balance or ion balance that exists across the membrane, okay. Now what happens at Donnan equilibrium, at the equilibrium state, okay, the rate of cation or the ratio of cations to anions, okay, in the inner side of the membrane is the same as the ratio of cations to anions on the outer side of the membrane, okay. This is at equilibrium, but a cell rarely exists at this equilibrium. Okay, what happens is that the cell, okay, has more and the cell membrane has an affinity to absorb anions from outside.
Yeah.
Okay, fine. So anions are absorbed, okay, or exchanged with cations from this. Okay, but then again, to maintain the ratio, further cations are taken in by the cell, okay, by other processes. Okay, getting? Again, so eventually your carbon and cation and anion ratio is almost towards the same level. Okay, anions are pulled in from here and cations are thrown out. Cations are further taken in by other processes. Okay, and anions, so they are anyway we add fertilizers and everything, and we are maintaining the level, right, of anions. Getting?
Yeah.
Harry, understanding? Okay. So this was about passive absorption, where there was no energy involved, nothing. Only with the ratio or wherever there's deficient, it was being compensated, balanced. Okay, basically balancing act over here. Okay. Now coming to your active absorption. Active absorption is basically when there is metabolic energy being used at the expense of the plant. Okay, energy produced by the plant is being used up in the process of taking up ions or taking up nutrients. Okay, when I'm talking about ions, I'm talking about nutrients as they've been broken down. Okay, now carrier concept. Simple. Okay, carrier, a molecule which helps to take in an ion from one end to the other end or transport it. Okay, now what this carrier concept says, okay, is that this space in a cell, okay, the space which is permeable to ions is limited. Okay, as in there will be some inner region, okay, in a cell, okay, which will be completely impermeable to ions. Okay, so if ions enter, okay, by whatever process, diffusion, ion exchange or any other process, okay, they enter in from here, okay, for the ion, okay, supposing it's a positively charged ion, to cross this barrier and come over here, it requires a carrier molecule. Okay, so this carrier molecule forms a reversible bond with this ion. It will move, transport the ion, dissociate from the ion, go back to where it was. Okay. So we are talking about the carrier concept. Okay. In the carrier concept, there is a carrier molecule, okay, which will attach to will form a reversible bond. Okay, and then will displace the ion to this side. Okay. So basically, what happens is that ions accumulate at this inner surface of the cell. Okay, then for them to be transported, they require a carrier. Okay, now how does the carrier know that it has to go and bind? Okay, so when ions accumulate over here, okay, two enzymes get activated. Okay, sorry, one enzyme gets activated known as phosphokinase. Okay, this phosphokinase along with ATP, okay, it will use an ATP molecule to activate this carrier. Then this activated carrier will go and bind to the ion. Then will release and the carrier will become deactivated. Okay, understood? Now the common carriers that are there, okay, in the plant cell is lecithin. Have you all heard of this? It's a protein. Okay, lecithin is a carrier. Then there is cytochrome. We all have heard of cytochromes. Photosynthesis, cytochromes we heard. Okay, so cytochromes are also carriers. Okay, then apart from it, the other carriers are also various other kinds of proteins. Okay, sometimes even proteins act as carriers, like the ones in your embedded in your which layer, which are acting as carriers, phospholipid bilayer in your membrane. Yes, okay, fine. Now what happens in terms of lecithin? Okay, now this theory or the lecithin carrier theory was proposed by Clark, okay, a scientist called as Bennett Clark. Okay, what they said is that lecithin, okay, has both cations and anions. The lecithin protein, okay, is made up of both types of charges, okay, cations and anions. So what they said is that lecithin carries cations and anions simultaneously, as in if an anion is present over here, okay, it will bind to a cation. Okay, if a cation is present over here, it will bind to an anion. Okay, getting? It will carry both the kinds of charges over here, put them, go back. Okay, so it carries both the types of charges or both types of ions simultaneously. Okay, then there is the cytochrome theory. Cytochrome theory, as we all know, cytochrome, whenever any reaction, okay, what kind of reactions are taking place using cytochromes? Your dehydrogenase reactions. Okay, so as and when the cytochrome reaction taking place, or when this is the ATP is being used in cytochrome reactions, okay, it's converted to ADP and okay. Now what these cytochromes do is that they take in cations, because remember when we were doing the cytochrome, this during non-cyclic photophosphorylation, okay, whenever cytochrome reaction takes place, an electron is given off. Electron is the one which is passing, right? So when this is passing the electron to something else, okay, in return it is taken a cation. Okay, then again when the process is starting, okay, again when an electron comes in, it gives off that cation. Okay, and while it's doing this process, it's traveling from in between that inner space of your cell. Getting how cytochromes are acting? Okay, now when this was a very brief this, okay, now let's see exactly what happens. Cytochromes get oxidized on one hand of the, or one half of the reaction, and the other half of the reaction they get reduced if electrons are being passed by. Okay, so on the outer surface, okay, on the outer surface, when the cytochrome travels to the outer surface, okay, of your cell, it gets oxidized. As it loses the electron, it gives off the electron, okay, and it picks up an anion. Okay, then when it returns back towards your inner side, your oxidized cytochrome, okay, it gets reduced because an electron is added. Okay, at the same time it dissociates with the anion it had taken. This process keeps on continuing of oxidation reduction, so basically a redox reaction occurring, okay, which in terms is going with our photosynthesis, which is also a redox reaction. So along with the photosynthesis, it's also doing this process of transferring of nutrients. Okay, then your second one is your electrochemical gradient hypothesis. Okay, see, both lecithin and cytochrome we're talking about, they are acting as carriers. Okay, now electrochemical gradient is very simple. What the electrochemical gradient hypothesis says is that there is a constant electrochemical gradient maintained, wherein the inner side of the cell, okay, or the inner mass of the cell has a tendency to always be negative in nature, okay, than your outer side. Okay, so because it has the tendency to be negative in nature, it's going to keep on taking in anions. Okay, so electrochemical gradient, easy enough. Okay, so we finished with the different processes, okay, that come. Passive absorption, active absorption, understood? Wherever we talk about active absorption, ATP is being used. Okay, then we talk about cytochromes. You all understood cytochrome well? Okay, if you all remember photosynthesis, you all will understand cytochrome theory. Okay, understanding?
Yeah.
Okay, now coming to the last part of this chapter. Okay, that is, we studied about all the different types of nutrients, but the main nutrient, okay, or what maximum is required by the plant is your nitrogen. Okay, so we're going to study how nitrogen nutrition takes place in plants. It's a very interesting process. Okay, as we all know, we've studied right from our childhood about only leguminous plants. But apart from leguminous plants, there are other nitrifying bacteria also present in the soil, okay, which do a lot of work. Okay, let's see. This is your TS of a root, okay, which has root nodules. Okay, let's come to this later. Okay, Starting with the nitrogen nutrition, okay, we all know nitrogen is the essential mineral nutrient required by the plant, okay. Now, why? Because it is a major or essential component of what all? You tell me. DNA, proteins, okay, cofactors, enzymes, all require nitrogen, okay. Now, the conversion of atmospheric nitrogen into organic compounds and further from those organic compounds into forms that can be directly taken in by the plants, this entire process is known as nitrogen fixation, okay. Now, nitrogen fixation can take place or can be done by free living bacteria in your soil, okay. Cyanobacteria are part of free living bacteria, or your root nodule bacteria which form a symbiotic relationship, okay. Whatever method, okay, both these will fix nitrogen in the same way. The basic mechanism in which they fix nitrogen is the same, okay. I mean the reactions that occur are the same, okay. Now, coming to your free living bacteria may be autotrophic or heterotrophic, okay. Autotrophic as in they use electrons to perform photosynthesis, okay, or heterotrophic they develop or they take in nutrients from the soil for the survival, okay, apart from your nitrogen which they take in from the atmosphere. Now your free living bacteria are your cyanobacteria like Anabaena, Nostoc. We've heard of all of these, okay. Anabaena is used very extensively as a fertilizer also, okay. Then are your root nodule bacteria. Root nodule bacteria, the most famous example is Rhizobium, okay, fine. Now, root nodule bacteria, okay, see these regions that you can see, okay, these are the regions in your root, okay, when you take the TS of a root where your bacteria has colonized, okay. The process or the reactions of nitrogen fixation, okay, take place in this, wherein the bacterioid, okay, the bacteria basically reside, okay, fine, understanding. Your infection thread, okay, or basically your infection starts in from here, okay, one root has been infected, then it goes in and colonizes wherever it has to, okay. Now, the legume, okay, see when the part of the root which is colonized by the root nodule bacteria is known as legume, okay. Root nodules and the root together is known as legume, okay. Now the legume provides carbohydrates to the bacteria, so in turn the bacteria, it breaks down these carbohydrates, okay, uses the energy, okay, which is produced by breaking down of oxidation of carbohydrates, okay, and this energy is used to break down your ammonium, okay, to forms which can be taken in by the plant, okay. But who makes this ammonium available to the bacteria in the first place? Okay, all your other free living bacteria, okay, getting. Now, biological nitrogen fixation, okay, occurs by a term, okay, or by a complex known as nitrogenase complex, okay. This is present in all organisms which are capable of nitrogen fixation, even in your root nodule bacteria, okay, and it is usually seen over here in the bacterioid region, okay. Now your nitrogen complex is made up of two parts, okay, or basically it has two regions, okay. First is your reductase, okay, which contains enzymes which reduce, okay, and then is your nitrogenase, okay, fine. Apart from it, the nitrogen complex also consists of molybdenum and ferrous. This, remember when we were studying molybdenum, we said it was required for nitrogen fixation, okay, fine. This molybdenum ferrous, they act as your coenzymes, okay, help in your reactions, okay. Now reductase, okay, it reduces whatever compounds are present, okay, carbohydrates or whatever, and provides electrons to your nitrogenase part. Your nitrogenase part is the one which converts it ammonia into various nitrate forms. Okay, fine, getting? Now this whole nitrogenase complex, okay, have you heard the term called as leg hemoglobin? Never? Okay, you know hemoglobin, the oxygen carrying pigment, okay, in our body. If you see, have you ever seen the root of groundnut or anything? These root nodules are pinkish or reddish in color. Okay, they'll be pinkish or reddish in color. Okay, this pinkishness is because of this pigment called as hemoglobin. Hemoglobin in our body is also red in color. Okay, it is the pigment which provides the redness to the blood, right? Okay. Now, why is this leg hemoglobin present? Okay, it is also a variant of your hemoglobin only, which is seen in humans, because this nitrogenous complex is very much sensitive to oxygen. Okay, a little bit of oxygen, okay, if it receives, it will disintegrate completely. But you can't prevent oxygen from coming in, because oxygen is freely available in the soil, even within the roots. Right? Oxygen is always being transported. Okay, so what this leg hemoglobin, okay, or this hemoglobin is always associated with the nitrogenous complex, okay. So whatever oxygen it sees in the surrounding, okay, it will absorb that oxygen, preventing oxygen from interacting with the nitrogenous complex. And anyway, it is a carrier of oxygen. And it is a mobile enzyme, okay. It will go and transport the oxygen somewhere else. Okay, fine. And that reddish or pinkish color is because when oxygen is bound to that hemoglobin, it gives that reddish color. Okay, fine. Now okay, so it's talking about the nitrogenous complex, okay, which has your reductase and nitrogenase. Okay, your molybdenum and ferrous protein is present in your nitrogenase complex. Okay, ferredoxin, aware of the protein ferredoxin, okay, okay, it carries electrons. Okay, now ATP is converted to ADP. It utilizes, okay, your ATP to make electrons dissociate from ferredoxin. These electrons are transported to your nitrogenase complex, where your nitrogen, okay, is converted to ammonia. Okay, fine, getting your this. Apart from molybdenum and ferrous, there is also cobalt, okay, which acts as a coenzyme or cofactor. Okay, cobalt is present in or is an essential part of vitamin B6. Okay, vitamin B6 is present in plants. Okay, fine. Now this ammonia, okay, some part of the ammonia, okay, which is taken or which is formed, okay, is directly used for in the synthesis of different amino acids. Okay, some of it has to be converted to nitrates. Okay, now we are coming to nitrogen converters in the soil. Okay, once ammonia is formed, okay, either by your root nodules or even by your other free living bacteria, okay, this ammonia has to be converted. Okay, so oxidation of ammonia to nitrate in the soil is done by mediation of two groups of bacteria. Okay, ammonia formed. Okay, first thing, nitrogen to ammonia, okay, done by nitrogenase complex. Okay, now ammonia to nitrates, okay, done by nitrosomonas. Okay, fine. Heard of this? Nitrosomonas and nitrobacter. Okay, both these will help in conversion of ammonia to nitrates. Okay, and they are also known as nitrifying bacteria. Okay, fine. Now, what nitrosomonas does, okay, is it converts your ammonia to nitrite, okay, and nitrobacter, okay, converts your nitrite to nitrate. Two-step process. Okay, fine. Clear? Okay, fine. This is done by okay, clear? Fine. Now, sometimes what happens is when excess of your nitrates are left, which is not taken in by the plant after some amount of time, okay, after a prolonged period of time when nitrate is not taken in by the plant, this nitrate is converted back to your nitrous oxide, okay, or to nitrogen, okay, and this process is known as denitrification, and the bacteria involved in doing that is known as denitrifying bacteria. Okay, fine. So, nitrate is then nitrification. I'll come later to the bacteria involved in denitrification. Okay, now nitrate is being formed. Okay, let's forget this denitrification part. We are still at nitrification. Okay, nitrate is being formed. It has to be taken in by the plant, assimilated somewhere in the plant. Okay, so now we are coming to nitrogen assimilation. Remember, we are still in the process of nitrogen fixation. Okay, your first part, second part done, third part done, now your fourth part. Okay, nitrogen assimilation. Okay, now nitrogen assimilation. Okay, roots of most higher plants. Okay, they absorb nitrogen from the soil in the form of nitrates, like we said before. Okay, now nitrogen, okay, in this form as nitrate, okay, cannot be used by the plant. It can be taken up by the plant, but now the plant has its own fuss. It will not use this in a nitrate form. So once into the plant, okay, your nitrate has to be converted into ammonia again. Okay, getting it clear? The roots, okay, not able to take in ammonia, so it has to be converted to nitrates. Once taken in the form of nitrates in the plant body, it has to be converted back into ammonia. Okay, getting it? So now this process, okay, of nitrate getting reduced back to ammonia, okay, is done by two. Okay, nitrate reductase, nitrite reductase. Again reverse. Okay, so NO3, okay, nitrate reductase, enzyme present where? Within the plant body. Okay, these enzymes are present within the plant body. Okay, clear? Okay, exactly reverse. Okay, this step is your fourth step, that is your nitrogen assimilation. Okay, this is done by enzymes which are present within the plant body. Okay, okay. Now your nitrate reductase, okay, utilizes NAD as a cofactor in this process. Okay, and also molybdenum. Okay, fine. This was the end of your nitrogen nutrition in plants and the end of this chapter, that is your mineral nutrition. Okay. Okay. So now we are coming to a new chapter, that is biofertilizers. Okay. Now, before in this chapter, before coming to the actual biofertilizer part, we are going to study about manures and chemical fertilizers. Okay. First of all, I want you all to tell me why are fertilizers required. Yeah, nutrients which are lost in the soil. Okay. Apart from that, okay, plants taking up nutrients. Some of the nutrients are also lost by erosion, leaching. Okay. So basically, soil is being constantly depleted of its nutrients. So we have to provide nutrients back when we want to grow crops. Okay. Right. Now, coming to your type, how you can replenish your nutrients. Okay, you can either add manures. Okay, manures are basically organic matter. Okay, which contains some portion of all the nutrients. Okay, wholesome meal to the soil, you can say. Okay, then there are chemical fertilizers and biofertilizers. Now, manures. Okay, when we add manures. Okay, first let me tell you what are manures. Manures are organic substances which are obtained by how decomposition of animal and plant wastes. Okay, and they are provided to the soil. Now, they will provide your, a little of each of the nutrients. But supposing your soil becomes deficient in one particular nutrient, then just adding manure will not help. You need to give large amounts of that one single nutrient. That's when your fertilizers come into picture. Okay. Now first coming to manures. Okay. We have different types of manures also. Okay. I think you all know all of these. Okay, you all are studying in school. The one only new to you all would be green manure. Okay. Farmyard manure, simple. All your agricultural wastes. Okay. Not kachra, but all your my God, like processing of your wheat bran, all this. Okay, oil cakes that are remaining, cattle feed. Okay, with leftover cattle feed. All that composed of your farmyard manure. Okay, it is good, but it does not provide potassium. Okay, one thing farmyard manure does not provide is potassium. So if your soil is healthy enough, okay, all you need to do is just add farmyard manure along with something known as they are deficient in phosphate, not potassium. Okay. Fine, superphosphates. Okay, all you need to do is just add superphosphates or phosphate compounds along with farmyard manure, and that will be enough. But that is when, in case your soil is already healthy. Okay, fine. Then composted manure. Simple, composting. Okay, all your waste, now your waste. Okay, you put it, okay, you let it decay, then same thing you apply to plants. Okay, then there is sludge and sewage. That is in liquid form. Okay, this is usually done, this sludge and sewage, okay, is usually, used during irrigation. When liquid manure has to be supplied during irrigation, in the irrigation water, your sewage is taken in, and because sewage contains, when you're in the process of collection and all, okay, your sewage and sludge contains a lot of matter which has already started decaying. Because of it, all your nutrients, okay, or your organic matter is readily available to the plant. Okay. Sludge is basically very thickened sewage. Okay, like sewage, okay, is like ones that is collected from your homes and all. Okay, sludge is when you have like concentrated amount of sewage that is released from industries. Okay, that is sludge. Okay, like sugarcane industry, okay, such places your sludges obtained, okay. Then is green manure. Okay, now what is green manuring? Basically, like the last lecture we studied about concept of leguminous plants. Okay, so what we do is we take either leguminous plants in combination with other fast growing crops, not crops exactly, fast growing plants which are not of much use to us, okay, as in we cannot extract anything. So before our growing season, before I have to sow the seed of supposing wheat, if I have a wheat farm, okay, before I sow the seeds of your wheat, okay, I will grow like around a month or a month and a half prior to it, I will grow your leguminous plant along with some fast growing plants. Then what I'll do is I'll plow the field, okay, so basically crush all these plants which I have grown and make it available to the soil. Eventually over a month's time that plant will start decomposing, it will become organic matter, okay. Cases where this is very helpful when your farms or something is very far away from any of your industrial areas where you will not get any of this, okay, fine. And leguminous plants obviously because they will replenish your nitrogen back into the soil, okay, clear? Now manures was very simple. And farmyard manure, cow dung doesn't come in sludge. Sludge is like the one which is produced as waste. Cow dung is farmyard, not cattle and all. That comes in your farmyard, farming process mein jo bhi, okay.
Only cow dung or any other dung?
Any cattle dung, farmyard manure, okay. Okay. Now coming to this concept wherein what I was telling you all, that when only a particular nutrient, okay, or a particular element is absent from the soil or is deficient, okay, we have to provide it externally, right? In such cases manures and all will not help, okay. So then comes the concept of fertilizers, okay, which are produced, okay, by industries chemically. So chemical fertilizers, okay. Now so I'll just tell you, fertilizers are which are generally inorganic, okay, that means rarely contain any organic matter from them but contain compounds which are rich in plant nutrients, are known as your chemical fertilizers, okay. Now coming to classification of fertilizers, depending on which nutrient they are supplying, okay, in the soil, you have your nitrogenous fertilizers, then you have phosphatic fertilizers, potassic fertilizers and compound fertilizers, which will be obviously mixture, okay, and mixed fertilizers, something known as mixed fertilizers also, okay, fine. So nitrogenous, then phosphatic, potassic, compound, mixed, okay, fine. Now coming to your nitrogenous fertilizers, okay, fertilizers which contain nitrogen as your primary, okay, that means around 50% or more of your compound will be made of nitrogen, but obviously nitrogen along with something else in the form of a nitrate, okay, fine. So now these nitrogenous fertilizers, okay, based on the other component that they contain, okay, or the secondary component that they contain, can be divided, okay, into four, that is your nitrates, okay, if there's go back to your last chapter, okay, when we see in the previous season stunted growth, certain
there is a lot of nitrogen present in the air also.
Yeah, but then plants cannot use atmospheric nitrogen. The whole concept of nitrogen fixation. Plants cannot use atmospheric nitrogen. They can only use atmospheric oxygen and carbon dioxide. Right? Okay. Whole nitrogen fixation is for the same thing, no? Because plants cannot use nitrogen from air. Okay. Yeah, so based on the form of nitrogen, there are 4 classes. Okay. Nitrates. Okay. Then there will be ammonium and ammonium salts. Okay. Then there will be chemical compounds which contain nitrogen in an amide form. Okay. NH2. Okay. And plant and animal byproducts. Okay. These are the 4 types of nitrogenous fertilizers. Okay. Fine. The most commonly used nitrogenous fertilizers are those of nitrates. Okay. Now let's see the different compounds that are used. Okay. Sodium nitrate, easily available, most frequently used. Okay. Now sodium nitrate contains about 16 to 20% of nitrogen, and the rest is sodium. Okay. 16 to 20% nitrogen and the rest is sodium. Okay. Now the reason why it is highly used is because apart from only in the soil, okay, it can be applied